You know, for decades, we've had the sequence of the human genome, our complete genetic blueprint. But here's the thing, being able to read all the letters and being able to actually understand the story they're telling, well, those are two very, very different things. So today, we're going to dive into a groundbreaking AI model from Google DeepMind called AlphaGenome, and it's finally starting to help us interpret our DNA like a real instruction book, which has some absolutely staggering implications for our health.
All right, so here's the big problem we're up against. For the longest time, we've known that only about 2% of our DNA actually codes for proteins, the literal building blocks of our bodies. The other 98%, it used to be called junk DNA, if you can believe that. We now know it's anything but. This huge non-coding region is actually the master control panel for our genes, and it's where the vast majority of variations linked to human disease are hiding.
Just let that number sink in for a second. 98%. I mean, that's a huge, unexplored territory inside every single one of our cells. The biggest challenge in modern biology has been this. When a single letter in that massive instruction manual is misspelled, how do you even find that typo? And maybe more importantly, how do you figure out what that typo actually does?
Okay, so this gets us right to the heart of the mystery. Understanding this non-coding DNA, this genetic dark matter, has been one of the biggest hurdles in all of science. Because this stuff isn't just a random string of letters. No, it's this incredibly complex, multi-layered regulatory system that's running the whole show.
So what's this non-coding DNA actually doing then? Well, think of it this way. If your protein-coding genes are all the individual light bulbs in a massive city, then the non-coding DNA is the entire electrical grid, the power stations, and all the thousands of switches that control which lights turn on in which neighborhood and at exactly what time. It's an unbelievably complex manual, and a simple misreading is where so many diseases get their start.
For years, scientists have been throwing AI at this problem, trying to get it to read this manual for us. But they kept hitting these fundamental roadblocks. The AI models they built were powerful for sure, but they were always forced to make these compromises that really limited how useful they could be.
Okay, to really get what these limits were, let's use an analogy. Imagine the genome isn't just a book, but a whole library, a massive one, filled with millions of books written in thousands of different languages. Before AlphaGenome, any AI trying to make sense of this library was stuck with two impossible choices.
So the first big trade-off, it was like choosing between a microscope and a satellite. Some AI models could zoom in with incredible microscopic precision and read individual DNA letters, but they could only see a tiny piece at a time, completely missing how a switch thousands of letters away might be affecting things. Other models could see the big picture, the whole satellite view, but the resolution was so low they couldn't spot a single critical mutation. Then there was the second trade-off, specialization. Some models were like a translator who's a world expert in, say, ancient Sanskrit, but can't read a single word of anything else. They were brilliant at one very specific thing, but totally blind to everything else going on. So the big question is, how do you solve this? How do you build an AI that can be both the satellite and the microscope? One that can understand all the different languages in that library, all at the same time.
And that's where AlphaGenome comes in. It's designed to be a unified, universal lens for reading our DNA. It's built to overcome those trade-offs by combining that long-range context with single-letter precision, and the ability to understand tons of biological processes at once. Basically, it's a tool that lets you see the entire library from space and then zoom right in to read a single word on any page in any language.
Okay, so how on earth does it do this? It really boils down to three key things. First, it reads a massive chunk of DNA, 1 million letters at once, so it can capture those crucial long-range connections. Second, it's what they call multimodal. It predicts thousands of biological functions at the same time, everything from gene expression to how DNA is packaged. And third, and this is the kicker, it does all of this with single-letter precision. It solves both of those dilemmas we just talked about.
All right, but does it actually work? Well, the data is, wow, it's pretty striking. They put it head-to-head against the world's best models, including those super specialized ones. And look at this, out of 26 different evaluations, AlphaGenome either matched or beat the competition in 25 of them. So this thing isn't just a jack of all trades, the results show it's a master of them too.
Okay, we've established the model is incredibly powerful, but what can it actually do for us? This is where things get really exciting. Let's look at how AlphaGenome can actually trace the precise chain of events that leads to genetic diseases, going from just a prediction to a deep, real understanding.
Let's look at a real-world example. There was a tiny deletion, just 4 DNA letters, that was known to cause a problem with how a gene's instructions get put together. Now, AlphaGenome didn't just predict that, hey, something's going to go wrong here. No, it predicted the exact outcome, that an entire section of the gene's blueprint would just be skipped, messing up the final protein. That's the kind of precision that helps you find the true root cause of a disease.
And check this out, because this next example really shows off its power to see that big picture. Here, we're looking at a mutation that's linked to a type of leukemia. AlphaGenome was able to predict the entire chain reaction. The mutation creates a new landing spot for a protein, which then flips a dormant genetic switch thousands of letters away. And that switch turns on a well-known cancer-causing gene. It sees the tiny local change, the long-range effect, and the final devastating impact all at once.
And really, the key takeaway here is something the researchers themselves said best. It's not just about predicting if a genetic change is bad, it's about predicting how it's bad. By looking at all these different biological processes at the same time, the model can actually piece together the complete story of how one little typo in our DNA can lead to a really complex disease.
So all of this really points to the future, because a tool this powerful doesn't just answer old questions, right? It opens up entirely new doors for medicine and for science itself.
So what's the big picture here? What does this mean for all of us? Well, for patients with rare diseases, it could mean finally getting a diagnosis by figuring out what a mutation in their non-coding DNA is doing. For medicine, it could help us design smarter, more precise genetic therapies that go right to the root cause of a disease. And for scientists, it's like a computational microscope, letting them run millions of experiments on a computer instead of in a lab, which could speed up discovery like crazy. We are really, truly entering a new era.
And that leaves us with one last kind of mind-blowing thought. For the first time, we're moving beyond just sequencing the book of life to actually reading and understanding the instructions inside. So the question is no longer just what our DNA says, but with this incredible new understanding, what will we choose to build?